Hello, Anderson Smith again, and today we're going to talk about the cell that represents the nervous system, the neuron. The nervous system consists of neurons. They look different, but they all have common features. We'll show that the neuron consists of dendrites, a cell body, and an axon. The dendrites receive information from other cells, and the axon gives that information to cells further down the line. We'll see that the axon is actually covered by a myelin sheath, a sheath that actually speeds up the flow of information within the neuron and also sort of insulates it because it is electrical activity. By the way, the myelin sheath isn't actually completely finished until about the mid-20s. So when a child is born, they have almost no myelin, and then over the first 2 decades, they actually develop myelinization for all of the neural cells. The information, when it passes through the nervous system, is electrochemical, and we'll talk about that. And each neuron has both a resting potential when it's not being activated and an action potential when it is. So the action potential is actually the information that's carried through the neuron. And the neuron will fire, or it won't fire. It's all or none. There's no gradual or graded firing. If it reaches its threshold, it'll fire all the way down to the end of the axon. Now this can be a long ways because there's one neuron is actually long, so it comes out of your spinal cord and goes all the way to your big toe. So that's a pretty long neuron. But if that fires, the dendrites in the spinal cord, if that fires, then it'll go all the way down to the axon terminals in the toe. So it's all or none. Now, and then we're going to talk about the synapse, which is how neurons communicate with each other across really a gap between the two cells, which is a chemical process. Now here's a picture of the neuron. It's actually a picture from your OLI readings. And you can see the dendrites surrounding the cell body, and this is where information actually is where the neuron is actually stimulated. So the axon terminals from other neurons go to the dendrites, and if that reaches a threshold, then the action potential will travel, as you see in the red arrow, it'll travel all the way to the axon terminals. You can see the myelin sheath covering the axon itself, and at the end of the axon, the other axon terminals, where information now will stimulate cells further down the line. There are literally trillions of connections between neurons in the brain. And then, of course, there are the connections with the nervous system, the peripheral nervous system outside of the brain and spinal cord. Now, here's the action potential. The resting potential for most neural cells is actually negative. It's about minus 70 millivolts. And that's when nothing is happening, there's actually a difference between the inside of the cell and the outside of the cell, negatively charged. If it reaches threshold, then it goes from minus 70 millivolts to plus 40 millivolts. And if that occurs, that action potential occurs, it will propagate down the axon all the way to the end. And then it returns in about a millisecond, it returns to the resting potential. Now, if it's all or none, how does the neuron code information about intensity? Obviously, I can tell if I have a light touch and I have a much larger touch. Some of that, of course, is the number of neurons that are stimulated. But the neuron itself can actually code information about intensity by when it can fire again. There's a very slight refractory period as it's returning down to resting potential. And then you can see it's actually below the 70 millivolts and slowly goes up to the resting potential. And if it's an intense stimulus, I can fire earlier in that period after the action potential. So it codes things by the frequency of firing. And here you see a half second, really about 1.5 seconds. At threshold, it's firing. If I have 160% of the threshold intensity, it's firing at a faster rate. Then at 270% of the threshold intensity is firing at even a greater rate. So the frequency of firing really is determined by the intensity of the stimulation on the neuron. Now, how does information get from the presynaptic cell to the postsynaptic cell, from the axon terminals of one cell to the dendrites of a later cell. It does that through a process at the synapse where things called transmitter substances are actually released from the presynaptic cell. They have vessels containing these transmitter substances. When the action potential reaches it, it releases them into the synaptic gap, and they stimulate the chemical stimulated reaction in the postsynaptic cell. And then there are enzymes that are released to sort of get rid of the excess transmitter substance so it can fire again. There are probably 100 different transmitter substances in the nervous system that do different things and have different functions. And we're identifying new ones pretty much every year. But the process is the action potential reaches the axon terminal, sort of a button-like, at the end of the axon. The action potential stimulates the release of transmitter substances into the gap between the cells, and those transmitter substances travel across the gap and stimulate the postsynaptic cell. And then the excess enzymes remove the excess transmitter substances from the gap itself. Now, like I said, there are many different transmitter substances. You have to remember that some transmitter substances actually inhibit firing. So you can have a transmitter substance that excites the cell, or you can have transmitter substances that inhibit the firing of the postsynaptic cell. Excitatory, inhibitory. An example of that is reciprocal inhibition. When I cause a muscle to move, sort of automatically the muscles opposite that movement are relaxed, so I can do the movement without putting stress on the other set of muscles. So when I excite a movement, other neurons are inhibiting the muscles opposite of that movement. Reciprocal inhibition. So inhibition is, again, a very important function in the brain in addition to exciting through action potentials. Here's some example neurotransmitters that are used at the synapse by neurons. And I'll just mention, these are the more common ones, and I'll just mention what they're characterized by. Acetylcholine is a transmitter substance that's primarily used at the connections with muscles. So when the afferent nerve comes out of the spinal cord, goes to a muscle group, and causes movement, that's acetylcholine for the most part. Dopamine is a transmitter substance that's very important in emotion and motivation. Endorphins are transmitter substances that are released when you have, for example, when you do heavy exercise, or when you have sexual orgasm, or when you eat spicy foods. GABA is the primary inhibitory transmitter substance that actually reduces the probability of postsynaptic cells even firing. Glutamate is a transmitter substance that's primarily found in the brain. In fact, 90% of the synapses in the brain use glutamate as a transmitter substance. And serotonin is a transmitter substance that's important in mood and appetite, and actually also in aggressive behavior. So different transmitter substances are used by different sets of neurons to do different things. Thank you.